Miners used to carry a canary into the mine, not because they cared about the bird, because the bird collapsed from invisible, odorless gas long before the miners did. It was an early warning system for a threat you couldn't see until it was too late. Your brain has a canary. Its name is vitamin B12. And I'm going to show you today that this canary is collapsing inside millions of people whose lab reports say the word normal in reassuring black ink. I'm Dr. Padda, and this is the third pillar of brain terrain, the micronutrients, the master switches and fine tools of brain construction. Get the fuel right, get the structure right, and you can still lose the brain if these run low. This is the most silent of the four failures, and in some ways the most important because it operates across your entire life, from the womb to old age. Let's start with what happens when B12 is severely deficient because it tells us how essential this molecule is. In infants with severe B12 deficiency, we see brain atrophy on MRI, delayed myelination. Remember myelin from last episode, seizures, and often lasting cognitive damage, even in cases where the brain's structure improves once B12 is restored. This is not subtle. Deprive a developing brain of B12 and it cannot build itself correctly. But severe deficiency is the obvious case. The danger, the canary, is what happens long before anyone would call it a deficiency at all. Here is one of the most important brain studies most people have never heard of. Researchers at Oxford followed community-dwelling older adults, people without cognitive impairment at the start, and tracked their brain volume with MRI over 5 years. What they found, the people with the lowest B12 levels, but still inside the normal range, lost brain volume fastest. The people with higher B12 lost it slower or held steady. The ones in the bottom tier had roughly 6 times the odds of accelerated brain shrinkage. Read that again. Not deficient by the lab's definition, normal, and losing brain measurably on a scan year over year. So how can a normal B12 be quietly shrinking a brain? The answer exposes something uncomfortable about how reference ranges are built. A laboratory normal range is not a range of optimal. It is a statistical description of the population being tested. It's built around what's average. And here's the catch. The average modern diet is heavily plant-based. By some estimates, around 70% of American calories come from plants. And plants contain essentially no B12. B12 comes from animal foods, so the normal range is anchored to a population that is, on average, running low. We have baked widespread mild deficiency into the definition of normal. The canary is dying at a level the lab calls healthy, because we set healthy by looking at a room full of dying canaries. This is why, at region.md, a B12 that reads normal doesn't end the conversation. It starts it. We look at where you sit within the range, and we look at the functional markers, the downstream signals that tell us whether your methylation machinery is actually keeping up. Let me show you why B12 matters this much, because it's not really about the vitamin, it's about the engine the vitamin runs. B12 is a required cofactor for methylation, one of the most fundamental operations in your biology. Methylation builds and repairs DNA, insulates nerves, manufactures neurotransmitters, and clears metabolic waste. When B12, working with folate, runs low, the engine sputters, and a compound called homocysteine builds up in the blood. Elevated homocysteine is not just a marker, it's associated with faster brain atrophy and higher dementia risk. And here's the hopeful part, because I never leave you only with the problem. In a landmark randomized controlled trial, older adults with mild cognitive impairment and elevated homocysteine were given B vitamins, tree, B12 folate, B6, and the treatment slowed the rate of brain atrophy, in some analyses by a striking margin, particularly in the brain regions most vulnerable to Alzheimer's. Let that land. This is a rare thing in medicine. An intervention that slowed the physical shrinkage of the human brain in a randomized trial, not with a billion-dollar drug, with the exact nutrients an animal food-based diet supplies, and a plant-heavy one doesn't. Now, I want to take this all the way back to the beginning of life, because the same nutrient that shrinks an old brain also shapes whether a young one is built correctly. If you don't have enough B12 to maintain a brain, how could you ever have enough to grow one? Researchers asked exactly this using one of the largest child development data sets in the world, the ALSPAC study out of Bristol. They looked at mothers' B12 intake during pregnancy and followed the children for years. The children of mothers in the bottom 10% of B12 intake during pregnancy, mothers who were, again, mostly within normal, had measurably worse vocabulary, worse speech development, and worse mathematics performance, and those gaps persisted for as long as the study followed them, into the teenage years. The study simply ran out of runway. We don't know that the gap ever closed. And children raised on strict plant-based diets in early life, even when later switched to omnivorous eating, have shown persistent cognitive deficits and lasting marginal B12 status. The window for building a brain is not forgiving. B12 is the canary, the easiest to see, but it flies with a whole flock, and nearly all of them are concentrated in animal foods. Vitamin D3, brain development, and dementia risk signaling. And note the cruel irony we'll pick up next episode. Avoiding sun and avoiding fat, or blocking cholesterol, all impair vitamin D. Preformed vitamin A, retinol, from animal foods. The plant precursor converts poorly in many people. Choline, the backbone of acetylcholine, the memory neurotransmitter, and of every cell membrane. Creatine, not just for muscle, a cognitive energy buffer for the brain, and typically low in those who don't eat meat. Carnitine shuttles fat into the mitochondria to be burned, concentrated in meat. DHA and EPA, the long-chain omega-3 fats that make up the structural fat of the brain, poorly supplied by plant-only sources. These are not optional add-ons. They are core inputs for myelin, synapses, cell membranes, neurotransmitters, and the mitochondria that power all of it. Strip the animal foods out of a diet, and you don't just remove some protein. You remove most of the raw materials the brain is physically built and maintained from. Do that across decades, and a slowly shrinking brain is not a mystery. It's arithmetic. Here's the uncomfortable, liberating truth of this episode. The difference between a brain that grows and a brain that shrinks can hide inside a lab value your doctor called normal. The canary can be collapsing while the report reads green. At Regan.md, we don't stop at normal. We look at optimal, your B12 and its functional markers, your homocysteine, the full micronutrient picture, and whether your methylation engine is actually keeping your brain in good repair, because these are among the most correctable factors in all of medicine, if you catch them before the damage compounds. We've now built 3 pillars: fuel, structure, nutrients. But there's a fourth force, and it's not about what's missing from the modern diet. It's about what we've added, the inputs that don't just starve the brain but actively set it on fire. That's next. If you want the full version of what I just covered, the actual studies, the mechanisms, and where the evidence is genuinely strong versus where it's still preliminary, I wrote it all down. It's a book called The Starved Brain, and the link is below. Every claim in it is referenced, so you don't have to take my word for anything.